Hello dear students, now in this lecture we are starting with our next topic of chapter respiration, that is respiratory balance shift. Respiratory balance shift, and it is a calculative phenomena, and it is possible to make calculation of the net gain of ATP for every glucose molecule oxidized. But in reality, this can remain only a theoretical exercise. Okay. So this, in respiratory balance shift, this calculation of ATP from one glucose molecule can be made only on certain assumptions, and that assumptions are first, that it is a sequential orderly functioning pathway with one substrate forming the next, and with glycolysis TCA cycle and ETS pathway forming one after the other. Second, NADH synthesized in glycolysis. NADH synthesized in glycolysis is transferred into mitochondria. And undergo oxidative phosphorylation. Next, third point, none of the intermediate in pathway are utilized to synthesize any other compound. Okay, so no any intermediate used in any other pathway to synthesize another new compound. And fourth point, only glucose is being respired. No other alternative substrate are entering in pathway at any of the intermediary stage. So these are the assumptions that when we consider, then only we can make this particular calculation. So assumptions are what? That it is a sequential orderly functioning pathway and forming the intermediates one after the other in glycolysis, TCA cycle and ATS pathway. Second, NADH oxidizes and glycolizes is transferred to mitochondria and undergo oxidative phosphorylation. None of the intermediate in pathway are utilized to synthesize any another compound, and only glucose is being respired. No other alternative substrate are entering the pathway at any of the intermediate stage. When we consider this assumption and calculate the particular ATP calculation from the glucose, then we can make a respiratory balance sheet calculation. So the calculation is as follows. So first we take a glycolysis and we calculate the ATP from the glycolysis. Now glycolysis, in this direct ATP are obtained, that are 4 minus 2, so 2 ATP, okay, gain, and through ETS from 2 NADH2, that is 6 ATP. So total 8 ATP are gained from the glycolysis. Second is link reaction. In a link reaction, pyruvic acid is converted into acetyl coenzyme A, and here 2 NADH2, 2 molecule of NADH2 are formed, and through ETS this 2 molecule of NADH2 give rise to 6 ATP, because 1 NADH2 give rise to 3 ATP. So here 6 ATP from the link reaction. And third is Krebs cycle. In Krebs cycle, if we talk about from 1 glucose, now be remember, here 2 pyruvic acid will be obtained in one glycolysis and 2 link reaction will occur. How many? 2 link reaction will occur. So from 1 link reaction 1 reduced to second link reaction, second reduced to 6 ATP. Now in Krebs cycle, from 1 Krebs cycle 3 NADH2, 1 FADH2 and 1 ATP directly are obtained. 3 NADH2, so 3 into 3, 9 ATP. 1 FADH2, so 1 into 2, because 1 FADH2 gives to 2 ATP, so these are 2, and this is 1 ATP which is obtained directly. So total 12 ATP are obtained from the 1 glucose, 1 Krebs cycle. But from 1 glucose 2 Krebs cycle will work. So this becomes 12 into 2, so 24 ATP. So this 24 ATP, this 6 ATP and this 8 ATP, if we combine, then 8 plus 6 plus 24 become 38 ATP. So 38 ATP is a total gain from 1 glucose. Okay, so this is the calculation or total yield of ATP, that is 38 ATP from the 1 glucose molecule, and that is a respiratory balanced sheet. So now the net gain of 38 ATP molecule occur during aerobic respiration of 1 glucose molecule. But in most eukaryotic cell, the net gain of ATP is 36 ATP. So now the question is, what is the mystery behind 38 or 36 ATP from one glucose. So now we have calculated 38 ATP when we have considered that the particular NADH2 from cytoplasm moving to the mitochondria. But now we understand what will happen. So this mystery of 38 or 36 ATP is solved by Shuttles System. It is solved by Shuttles System. Suppose this is a cell, and in cell this is a mitochondria. Now what happens? Glucose convert into pyruvic acid, and this EMP or glycolysis pathway occur in cytoplasm. Okay. In cytoplasm, two NADH2 molecule are also produced. Now this NADH2 passes to the what? Now we know that ETS is located here in case of inner mitochondrial. Now NADH2 till will not enter this ETS, they will not give rise to 3 ATP. So they have to break through the ETS, pass through the ETS, their energy, and the 3 ATP will be synthesized. So to carry out this, they have to reach the mitochondrial matrix. Now to reaching this mitochondrial matrix, they use this shuttle. Now it is known thus, if this enter through this shuttle, this NADH2 will be equal to 3 ATP, and if this enter through this shuttle, the NADH2 is equal to 2 ATP. Now if we consider this shuttle, then NADH2 is equal to 3 ATP, then total calculation will be 38. But when we consider this NADH2 is equal to 2 ATP, then total 6 ATP will not be calculated. Here we calculate 4 ATP. So when we calculate 4 ATP in this, then the total calculation becomes 36. So how this is happening? How this shuttle system is working? So we talk about next topic, that is a two type of shuttle systems. So next we start with the shuttles or shuttle system. Okay. So what this shuttle system says? Shuttle system says that the NADH2 molecule produced during glycolysis cannot enter mitochondria. Directly. So they hand over their energy. So NADH2 hand over their energy to other molecules. This entry of glycolytic energy, this entry of glycolytic energy inside mitochondria is performed using specific shuttles, and there are two type of shuttles. Okay, first is malate aspartate shuttle, and second one we will talk about the glycerol 3-phosphate shuttle. Now in malate aspartate shuttle, if we study, so when we study this malate aspartate shuttle, we know that the cytoplasm is the region where glycolysis occur. Then when this NADH2 of cytoplasm need to enter mitochondrial matrix, it has to pass outer mitochondrial membrane, then perimitochondrial space, then inner mitochondrial membrane, and then mitochondrial matrix it will reach. Now we know that during cytoplasm, when glucose break into pyruvic acid, this NADH2 is going to produce. Now this NADH2 break into NAD plus and react with oxaloacetic acid to form malate or malic acid. Okay, so when an oxaloacetic acid react with the NADH2, that will give rise to NAD and malate. Now oxaloacetate cannot pass this membrane, so malate can pass this membrane. Now after reaching this, malate will break again. It will release its H plus, that is received by again NAD plus, and that NAD plus will be converted into NADH2 again. So here you can see NADH2 breakdown was occurred and malate was produced. When malate get broken up into the mitochondrial matrix, again NADH2 will be produced. So the energy of NADH2 of glycolysis passed in the form of NADH2. Now this malate will convert into oxaloacetic acid, but oxaloacetic acid cannot pass, so it react with NH2 and form aspartate. Now this aspartate can pass through this, so aspartate will come here, and this aspartate, when release NH2, it will form again oxaloacetic acid. Okay, so this is the way how the whole malate aspartate shuttle is going to function. And in malate aspartate shuttle, the NADH2 is giving energy in the form of NADH2. So if glycolytic NADH2 is ultimately transferring its energy in the form of NADH2 in mitochondrial matrix, means glycolytic NADH2 is equal to 3 ATP. And when we consider glycolytic NADH2 is equal to 3 ATP, total ATP calculation occurs 38 ATP from one glucose molecule. Okay, so this is the thing. Now this malate aspartate shuttle occur in heart, kidney and liver cells, and 38 ATP are obtained. Now we talk about another shuttle, that is a glycerol 3-phosphate shuttle. So second shuttle is glycerol 3-phosphate shuttle. In this, if we draw, suppose this is a cytoplasm, this is outer mitochondrial, if we cross the membrane of mitochondria, this is perimitochondrial space, this is inner mitochondrial membrane, and these are the mitochondrial matrix. Now if we see here, what is going to happen? In cytoplasm, glucose is going to break into pyruvic acid and it is going to produce NADH2. Now this NADH2 actually react with DHAP to form glycerol 3-phosphate and NAD get produced. Okay, so when it react with the DHAP, glycerol 3-phosphate is produced. Now in inner mitochondrial membrane, there is an enzyme complex known as succinate dehydrogenase complex. So now when this glycerol 3-phosphate passes, then they passes its particular energy to the FAD here, and the FADH2 will be produced, and here we get a DHAP again back. That DHAP will be used again here. So in this, when glycerol 3-phosphate shuttle is active, glycolytic NADH2 transferring its energy into the FADH2. So here by this shuttle, glycolytic NADH2 is equal to FADH2, and FADH2 is equal to 2 ATP. So when this shuttle is active, glycolytic NaDH2 is equal to 2 ATP and total ATP become 36 ATP from one glucose when we carry out a complete calculation. Now this shuttle occur in or found in flight muscle of cells and brain and in most of eukaryotic cell. This shuttle are absent in prokaryotic cell. This shuttle system is absent in prokaryotic cell, that you should remember. So this is the way how 36 and 38 ambiguity can be recovered or can be solved. Next we discuss efficiency of aerobic respiration. Now when we talk about efficiency of aerobic respiration, it is known that 1 ATP yield 34 kilojoule energy. The total energy obtained from, so total energy from 38 ATP is equal to 38 into 34, so that become 1292 kilojoule energy. Now one glucose stores 2870 kilojoule energy, but how much we get extract in the form of ATP, that is 1292. So efficiency is 1219 kilojoule, that is equivalent to 38 ATP out of the total energy present in glucose is 2870. And if we carry out efficiency, this FAT comes near about 45%. So whatever the energy which is stored in glucose, out of this only 45% of energy get released in the form of ATP from the one glucose, that is the efficiency 45% of glucose. Now we talk about the next topic, that is fermentation. Fermentation is a kind of anaerobic respiration. It is a kind of anaerobic respiration because first glycolysis happen and this fermentation will be carried out. It is carried out primarily by fungi and bacteria. Although people had been using the process in the preparation of wine since prehistoric time, but failed in this their attempt to understand the alcoholic fermentation. So this fermentation carried out by fungi and bacteria, Lactic was the first to discover fermentation. But Louis Pasteur concluded that fermentation occurred only when the living cell or living yeast cell were present and called this zymosis or fermentation. So what this Louis Pasteur concluded, that fermentation occurred only when the living yeast cells are present. Now this fermentation are of different type, and first we tries to study in this, that is a alcoholic fermentation. Now in alcoholic fermentation, if you study, this is the most common type of fermentation. This alcoholic fermentation, it is the most common type of fermentation taking place in yeast and certain bacteria. Be remember, alcoholic fermentation takes place in yeast and certain bacteria. This alcoholic fermentation takes place in two steps. It takes place in two steps. In first step, the pyruvic acid, that is CH3COCOOH, which is a keto acid. The two molecule of this pyruvic acid is obtained from the one glucose molecule. Now this pyruvic acid, in the presence of pyruvate decarboxylase and in the presence of enzyme pyruvate decarboxylase, by using TPP and magnesium as a cofactor, convert into CH3CHO and 2CO2 molecule. This is the acetaldehyde. So acetaldehyde molecule is formed here. After acetaldehyde formation, in second step, this acetaldehyde react with NADH2, and that is the two molecule of NADH2 which was synthesized in the glycolysis. So when acetaldehyde molecules react with NADH2, they accept H plus in the presence of enzyme alcohol dehydrogenase. So NADH2 breakdown occurs, and in alcohol dehydrogenase presence, NADH2 will break into NAD and H plus. H plus are received by this, so it will form 2C2H5OH plus 2NAD plus, and this C2H5OH is nothing but a ethyl alcohol. So such a way, in this two step, the ethyl alcohol will be formed, that is C2H5OH. So overall equation become like this, in which C6H12O6 in the presence of zymase complex convert into C2H5OH plus 2CO2 plus 58 kilocalorie energy. Okay, because NADH2 which was synthesized in glycolysis, because glycolysis is common to the aerobic and aerobic respiration. So NADH2, 2NADH2 which was synthesized in glycolysis get used here. So only 2 ATP net are obtained, that is equivalent to something 58 kilocalorie. One of the important point you should remember that in fermentation by yeast, when percentage of alcohol in sugar solution is about 13%, yeast cells get killed and fermentation stops. So a maximum the alcoholic concentration in sugar solution that can be tolerated by yeast is a 13%. Okay. Second type of fermentation is lactic acid fermentation. In lactic acid fermentation, pyruvic acid formed at the end of glycolysis, that pyruvic acid is converted into lactic acid by homofermentative bacteria. Homofermentative lactic acid bacteria that are Lactobacillus lacti. Okay, and during a lactic acid fermentation, this pyruvic acid, that is CH3COCOOH, react with NADH2 of the obtained from the glycolysis. Means here we can say these are the two molecules and two NADH2 will be reacted, and in the presence of lactate dehydrogenase and FMN and Zn2 plus, it convert into C3H6O3, a two molecule of this, that is a lactic acid. Now during this conversion or lactic acid fermentation, no CO2 evolution, that you should remember, but two NADH to get used. Third type of fermentation is lactic acid and ethyl alcohol fermentation. Lactic acid and ethyl alcohol fermentation. This both fermentation can carried out by only one type of bacteria, then this type of bacteria known as heterofermentative bacteria. So both lactic acid and alcoholic fermentation simultaneously carried out by the heterofermentative bacteria. So here CH3COCOOH, that is a two molecule of pyruvic acid, convert into C3H6O3, that is lactic acid, plus C2H5OH, that is ethyl alcohol, plus CO2 in presence of heterofermentative lactic acid bacteria, which will synthesize lactic acid along with the ethyl alcohol. Fourth type which we are going to discuss, that is acetic acid fermentation. In acetic acid fermentation, pyruvic acid, that is CH3COCOOH, first in presence of Acetobacter aceti, converted into CH3CHO, that is acetaldehyde plus CO2. Now this acetaldehyde, that is CH3CHO, on reaction with water and by the removal of 2H plus, it convert into CH3COOH, that is acetic acid. So in such a way acetic acid can be produced. So because of this it is known as acetic acid fermentation. Okay, so these are the different types of fermentation, but in this be remember, dear students, two are more important, that is alcoholic fermentation and lactic acid fermentation. So after studying this fermentation, we carry out comparison of fermentation and aerobic respiration. Fermentation and aerobic respiration. And in comparison, first we understand that there is a partial breakdown of glucose, partial or incomplete breakdown of glucose occurs in fermentation. Whereas in aerobic respiration, complete oxidation of glucose, in aerobic respiration, glucose is completely degraded to CO2 and H2O. Second is net gain if of only 2 ATP, net gain of only 2 ATP in fermentation, whereas it is very high of 38 or 36 ATP in aerobic pathway. And third comparison is NADH is oxidized to NAD, that is NADH to break down to NAD is rather slowly during fermentation and very vigorous or fast during aerobic respiration. Okay, so in aerobic respiration it is fast, whereas it is slow in case of the fermentation. So these are the comparative analysis of ferment and aerobic respiration, and here aerobic respiration and fermentation are over. Next we are moving to the another topic, that is amphibolic pathway. Next topic which we are going to discuss, that is a amphibolic pathway. Amphibolic pathway are those pathways, these are those respiratory pathways which involve both catabolic and anabolic pathway. So those respiratory pathways which involve both catabolism and anabolism, they are known as amphibolic pathway. Now if we see the pathway which we usually discuss in the breakdown of glucose, that we know that initially we have a carbohydrates, and these carbohydrates break up into the glucose, and that glucose is converted into the DHAP and PGAL. Okay, then DHAP and PGAL are produced. This DHAP and PGAL isomer, this PGAL will form pyruvic acid, as we know that this pyruvic acid will form acetyl coenzyme A, and acetyl coenzyme A will carry out a Krebs cycle. So this is a normal breakdown of the glucose. But when this glucose store get complete in cells, the body will be dependent on the fat, and when body will be dependent on the fat, it will break into fatty acid and glycerol. It will break into glycerol and fatty acids. Now this glycerol will be converted into dihydroxyacetone phosphate, and through this they will enter this pathway. Through this particular DHAP they will convert into PGL, pyruvic acid, and will carry out a Krebs cycle, and they will get the energy. Body cell will get the energy. Now this fatty acid convert into acetyl coenzyme A by a process of beta oxidation, and through acetyl coenzyme A it will give the energy. But what when fatty acid and glycerol are also over? Then body will be dependent on the proteins. means it will shift to the protoplasmic respiration, and proteins are broken into amino acids, and this amino acid will convert into pyruvic acid, and this pyruvic acid will undergo acetyl coenzyme and Krebs cycle will be formed and cell will get energy. Means the acetyl coenzyme is the common link between acetyl coenzyme A is a common link or intermediate in breakdown of carbohydrate, fat and protein. First important point is this. Now here we are seeing that catabolism is happening and all the respiratory intermediate are forming through the breakdown, but it is not like that always catabolism will occur and this respiratory intermediate will form and we will get energy. Some of the respiratory intermediates of this pathway are also used in synthesis of some of the another compounds, means they are involved in anabolism of some another compound as well. So what they are? So if they are involved in anabolism as well, then means they are the part of amphibolic pathway. So next we talk about some of the intermediate and some of the intermediates of this various pathway acting as a precursor for the another pathways. So various intermediates of the pathway are precursor of various compounds. And if we take an example, then we come to know that first is acetyl coenzyme A. Now this acetyl coenzyme A is a raw material for chlorophyll. Acetyl coenzyme A is a raw material for carotenoids, then terpenes and gibberellins. Okay, these chemicals can be synthesized from acetyl coenzyme and it is also formed by the breakdown, so it is a part of anabolism and catabolism. Second, succinyl coenzyme A. This succinyl coenzyme A is acting as a raw material for chlorophyll and cytochrome. Third is oxaloacetic acid. This oxaloacetic acid is a raw material for alkaloids and pyrimides. And fourth is alpha ketoglutaric acid, that is acting as a raw material. Alpha ketoglutaric acid acting as a raw material for amino acid synthesis. Means these are showing that they are a precursor of different component and they are synthesized by breakdown also, means they are the part of amphibolic pathway. Next pathway which we discussed, that is hexose monophosphate shunt or direct oxidative pathway. It is also known as PPP means pentose phosphate pathway. Okay. Now, in addition to the normal process of oxidation of carbohydrates through the glycolysis and Krebs cycle, there is another process by which plant could oxidize carbohydrate to obtain the energy. So this is the alternative pathway to glycolysis and to obtain the energy. Okay, this pathway occur in cytoplasm and chloroplast in presence of oxygen. Okay, it is found in both prokaryote and eukaryote. Okay, and it is also called as Warburg Lippmann Dickens pathway. It is also known as Warburg Lippmann Dickens pathway, which got discovered by the scientists. Now, some important steps of this pathway. In this, first what happens, glucose is oxidized to the glucose 6-phosphate. So first what happens, if we have a 6 molecule of glucose, it will react with 6 ATP and produce 6 molecule of glucose 6-phosphate. Along with 6 molecule of glucose 6-phosphate, it will produce 6 ADP. That is the first step. In the second step, this glucose 6-phosphate, that is the 6 molecule, will react with 6 water and 6 NADP plus, and in the presence of glucose 6-phosphate dehydrogenase, in the presence of glucose 6-phosphate dehydrogenase, this glucose 6-phosphate, by reacting with this, it convert into 6 phosphogluconic acid, and that is a 6 molecules plus 6 NADH are obtained. 6 NADPH are obtained. This means here 6 NADP are used, okay, in this process. Then in third step, 6 phosphogluconic acid, 6 molecule of this, react with again 6 NADP and give rise to 6 molecule of ribulose 5-phosphate plus 6 CO2 plus 6 NADPH2. So 6 NADPH2 are formed in third reaction, 6 NADPH2 are formed in second reaction, and 6 CO2 are released. This ribulose 5-phosphate 6 molecule of ribulose 5-phosphate, which is a 5-carbon compound, get converted into a 5 molecule of fructose 6-phosphate, that is 6-carbon compound. Total carbon will be same. Here also 6 into 5 30 carbon, here also 5 into 6 30 carbon. Now this fructose 6-phosphate get isomerized into glucose 6-phosphate, and initially we have used 6 glucose 6-phosphate that get regenerated, 5 glucose 6-phosphate that get regenerated. So 1 from the new glucose and 5 regenerated will use, and total 6 glucose 6-phosphate enter this pathway, and this pathway is continued by this all the steps which we have discussed. So in this 5 molecule are regenerated, one is used new. So in this 6 CO2 are released and 12 NADPH2 synthesis occur. Now 12 NADPH2, 1 NADPH2 equal to NADPH2 is equal to 3 ATP if I consider, then 12 NADPH2 is equal to 12 into 3, that is 36 ATP. But 1 ATP is used in first step, so minus 1 ATP, so total it become 35 ATP. Total it become 35 ATP. So thus in terms of energy output, the hexose monophosphate system is almost as efficient as aerobic pathway, because in aerobic pathway also we have studied up 36 or 38, and here also we are getting 35 ATP, means it is almost same. This pathway is important in tissues like liver, adipose, germinating seeds, and ovary. Okay, and this pathway acts as a safety valve or shunt to EMP. That is important point, because when there will be a deficiency of enzyme or anything can be there, at that time this pathway help us to get the energy, and it is very low activity. It has very low activity in skeletal muscle. Okay, so this is the whole pathway, alternative pathway to the glycolysis or aerobic respiration of glucose, that is hexose monophosphate shunt or pentose phosphate pathway. Now we are coming to the next topic, that is respiratory quotient, and it is the last topic. So next topic is respiratory quotient or respiratory ratio. This respiratory quotient can be written as RQ. So respiratory quotient is equal to volume of CO2 released upon volume of O2 consumed. So if we take a ratio of CO2 evolved to the O2 absorbed, then this is known as the respiratory quotient. So it depends on the type of respiratory substance. This respiratory quotient depends on type of respiratory substrate. This is measured by the help of an apparatus known as Ganong respirometer. It is measured by instrument known as Ganong's respirometer. Ganong's respirometer. And RQ may be equal to unity, more than unity, or less than unity. That is the depending on the substrate utilized. Now first we are taking some of the example. First, suppose respiratory substrate is carbohydrate. Respiratory substrate is carbohydrate. So in case of carbohydrate, the reaction is C6H12O6 plus 6O2 give rise to 6CO2 plus 6H2O. So in carbohydrate, if we talk about the RQ is 6CO2 upon 6O2, means it come to the 1, that is unity. Okay. So when a plant material where carbohydrate serves as a respiratory substrate, then the respiratory quotient becomes 1. So example are germinating seeds of cereals, leaves kept in dark, flowers and fruits rich in carbohydrates have a RQ is equal to 1. So when carbohydrate is respiratory surface, respiratory quotient is 1. When respiratory substrate are fat, the reaction look like this, that is a C51H98O6 react with 145 O2 and give rise to 102 CO2 plus 98 H2O. So if we take, this is a tripalmetine, that is fat. So RQ become 102 CO2 upon 145 O2. So it become 0.7. It become 0.7. So RQ of fat is less than 1, that is 0.7, and it is found in case of germinating fatty seeds like castor, mustard, linseed, etc. So the important point is that you remember that fat are poor in O2, so they need more O2 from outside. That's why the RQ become less than 1. Next we talk about RQ of third. When proteins are a respiratory substrate, the RQ become less than 1 here also. So in proteins also RQ is 0.9, that is less than 1. Okay. So example is germinating pulse seeds which have a protein and they are RQ is less than 1.9. Fourth is in succulent xerophyte. Now succulent xerophyte are those plants which hold the water, and in this if we consider during night, the stomata are open, so they exchange the particular during the night. So when we take a reaction C6H12O6 plus 3O2 give rise to C4H6O5 plus 3H2O. So during night when stomata are open, during day the oxidation particular does not occur, light is not, stomata are not open, so CO2 is not evolved, only O2 is used. So if we take a RQ, then 0 by 3, so it become 0. Okay. And next after fifth we consider in case of organic acid, then in case of organic acid it is greater than 1, and it is for malic acid it is 1.33, and for oxalic acid it is highest, that is 4. So these are the RQ for different organic acids. In case of anaerobic respiration, if we consider, then the reaction becomes C6H12O6. In the presence of gymase, it gives rise to the C6H5OH plus 2CO2, means O2 are not used. So RQ become 2CO2 upon 0O2. So this become infinity. So in anaerobic respiration, RQ become infinity. Then we talk about RQ of maturing seed, fatty seed. RQ of maturing fatty seed is greater than 1. Then RQ of mixed diet is 0.85. Then RQ of starred leaves is less than 1, and RQ of colored petals is less than 1. So these are the different RQ found with respect to the different respiratory substrate.